The next section in chapters 6 and 7 is to talk about how genes work. So we've already talked about what a genome is, what genes are, what chromosomes are. Now we're going to talk about the actual how do we look at the gene, get the information from the gene, and then read it. So we're just going to look at it as an overview, big picture. If you want, there are more details in your textbook, but we're just going to be looking at it big picture for this section of the chapter. A couple of terms before we get into talking about how the actual process works. So first, there's the genotype. So geno for genes or genetic. So the genotype is all of the genes that are contained in an organism. So this is all of the genes in the genome, right? It also is the genetic makeup of the organism. So the genotype is basically the genes, the directions section. You can have different sets of the directions because you can have those different alleles, and we'll talk more depth in the inheritance chapter, chapter 9, on how you can have different combinations of those alleles to get different phenotypes. Phenotypes are the physical manifestation of the instructions. So what we're going to be looking at here is we're going to be looking at how we read the genotype, the gene, to then get the phenotype, the physical material. In this case, we're going to be looking at a protein. All right. So if a person has one normal and one mutant copy of this gene, the BRCA gene, which is related to breast cancer, that describes what? So is that going to describe alleles? Is that going to describe traits? Is that describing their genotype or their phenotype? So those are all your different options there. So if they have a normal copy and a mutant copy of this BRCA1 gene, that describes their blank. If the mutation in the BRCA1 leads to breast cancer, that describes their what? And so here the answer is C, because the first part's talking about the genes, the genetic makeup, and then the phenotype is the physical manifestation. That mutation in the gene leads to breast cancer, leads to the changes in the cell. So that's the phenotype. Okay. All right. So what we're going to be looking at here as far as understanding how we go from the genotype, the genes, to the phenotype, the physical manifestation like a protein, we're going to need to look at the central dogma. So the central dogma is this, and we're going to add to it, but it is, we're going to go from DNA to RNA to proteins, right? And it's one-way street for what we're looking at. So the information flows from DNA to RNA to proteins. And we'll add some pieces and parts in as we go along, but that is the main idea here. Okay, so central dogma starts with the DNA. So we're going to start with DNA. And if we're working with a eukaryotic cell, where are we? If we're working with DNA in a eukaryotic cell, where in the cell are we at? We're in the nucleus, right? Because that is where the DNA is going to be found for a eukaryotic cell. So we've got our DNA, we've got our genes, because that's the information that we're going to read. So you can see here that this is in your book talking about this like your grandmother's cookbook. I don't know about your grandmother, but my grandmother does not allow me to take cookbooks out of her house. And it's very much like this for the cell, right? The cookbook or the genes cannot leave the nucleus. So if the cookbook can't leave, what can you do? We can make a copy, right? And that's what's going to happen with the central dogma, with how genes work. What you're going to have happen is you're going to get an enzyme. In this case, you can see it's called RNA polymerase. It's going to come in and it's going to make mRNA. That might sound familiar from back in chapter 3 when you learned about nucleic acids. The mRNA is a copy of the gene from the DNA, okay? And that copying process is called transcription. To transcribe something means to copy something, right? To write it down. And so this would be like writing down the recipe out of Grandma's cookbook so that you can take it with you, right? Because the cookbook can't go, but the copy can. So going to make mRNA through the process of transcription in the nucleus. Once the mRNA is made, the DNA is going to get closed up, so the gene will get zipped back up. But that mRNA can leave the nucleus. DNA can't, but the mRNA can. So now we've got our copy of the directions through transcription. mRNA is going to be able to come out to the outside of the nucleus, and it's going to be hanging out here in the cytoplasm. Now, the directions for mRNA are going to be to make something like a protein. So if we're going to make a protein, what is the mRNA going to need to come out here and find? What structure? It's going to need to find a ribosome, right? And so that's what our mRNA has found. It's found a little ribosome friend, because remember, ribosomes are protein factories, right? Now the ribosome could be hanging out in the cytoplasm. The ribosome could also be attached to the rough ER. As long as it finds a ribosome, that's fine. Now the ribosome is going to work to help figure out this information, okay? But it's going to need some help, because this cookbook, Grandma's cookbook, my grandma's cookbooks are written in German. I don't know about yours. And let's say I don't speak German. What am I going to need to do? I'm going to need to translate it, right? Well, the same thing is happening here. mRNA is a nucleic acid, right? Just like DNA. So it is written in nucleic acid speak. It's written in nucleotides, the A, T's, C's, and G's, or in this case, because it's mRNA, it's going to be written in A, U, C, and G, right? The ribosome, that guy speaks protein, speaks in amino acids, because that's what it works on to help build. So it's going to need a translator. And if you remember from the nucleic acid video that you saw back in chapter 3, it's going to use these little guys right here, tRNA, right? tRNA, transfer RNA, or you can think of them as translation RNA, but they're called transfer RNA because what they do is they can talk to the mRNA, because they speak nucleic acid, they speak nucleotide, but they also speak amino acid. Each of these guys has on the other end, you can see here, an amino acid. So they're coded to match up with the mRNA with specific sets of letters to put in the right amino acid. And so these guys come in, the tRNAs come in and translate. And so that's why this is called translation. It takes the information from the nucleic acid and turns it into protein, turns it into amino acids. Eventually, this entire mRNA will get read. So this is kind of like making the cookies, right? until you get your cookies, aka your protein molecule. And so that is how genes work overall. So we go from transcription in the nucleus, where we make that copy of the mRNA from the DNA. The mRNA goes out, finds its ribosome friend with some tRNA's help, translates through translation the directions from the mRNA, from nucleic acid, into amino acid to build the protein. Okay. So let's go back over here to our central dogma. That arrow is not supposed to be there yet. So remember, we started with our DNA, then went to RNA, then went to proteins. So now let's fill in some. We've now got transcription. Transcription is the step between DNA and RNA, and that happens in the nucleus, right? Then translation is the step between RNA and proteins that happens at those little ribosomes, because we go from nucleic acid, the mRNA, to proteins using those tRNA. So that is the overview on how these genes are read.